do the second half of our first morning session, and I'm going to introduce an undergraduate to you, Makenzie. Right being held before on the right page here. Makenzie Burrows, who's an undergraduate at the University of South Florida, will tell us a little bit about genomic variation of the Chelonid, Fibropapillomatosis-Associated Herpesvirus. I gotta stop reading these titles.
Take it away, thank you. So I am going to be presenting on the genetic variation of the Chelonid Fibropapillomatosis-Associated Herpesvirus. And I am an undergrad at the University of South Florida, St. Petersburg, working in the Break Art Lab at the USF College of Marine Science. So first off, I'm going to walk you through everything so that you understand the title of our presentation. So first off, we have chelonids. So chelonids are sea turtles. Right now, we're going to be focusing on 3 specific species: the green, loggerhead, and Thames ridley. And all these sea turtles are actually endangered or threatened, which is very important that we help keep them alive. So the reason for that is that they control jellyfish populations. They actually eat them. Why would you want to eat them? I don't know. They do. They also are the lawnmowers of the sea. So they actually eat the seagrass off the seafloor. And you think, oh, well, manatees do that, but they don't do it as much as sea turtles do. And that actually helps control those seagrasses and make them live. So if they don't get trimmed, they will die. And they're very important for food chains. Also, they actually have their eggs on the beach, and those eggs, when the sea turtles come out, there's still the yolks left in there. Or if they don't develop properly, they provide vegetation for that dune vegetation, which holds on that sand on the beach. So what is fibropapillomatosis, or FP, the easier way to say it? It is actually a neoplastic disease, meaning that it is tumor causing. So you can see up there that there's a giant gross tumor on the face of that sea turtle, which is really sad. And this is found worldwide in a circumtrophic distribution, meaning that it is in the tropics of the ocean. Also, it affects all 7 different species of sea turtles, but we're primarily affecting the green sea turtles. It is also discovered during the Industrial Revolution. These tumors on the turtles can range anywhere from 0.1 centimeters all the way up to greater than 30 centimeters in size, which is really large, like you can see on this one. And there can be 1 tumor to a ton of them all over the body. They can also be found internally. When they're internal, they're called fibromas, and those can actually be very deadly, but they're only found in less than 10% of the sea turtles that are infected. And then also their external masses. So the prevalence of FP varies by location. You can see this in this study. So they did a study on Florida, where on the East Coast there's much more prevalence of the disease in the inner coastal areas where there's more pollution and eutrophication, and that affects the disease and makes it more prevalent in the sea turtles. There's over 50% in this area here. And there's also, as it further goes out into the environment, into the ocean area, there's less prevalence, so it's less than 1% in the outer region. More about FP, we have its growth. So a 0 to 3 scale is what we use to identify FP. 0 meaning there are 0 tumors on it, and 3 meaning that there are tons of tumors all over the sea turtle or also inside of it. There's also regression. So if there is not a 3 status of this FP, then it will regress in the environment. And that is seen if you take the samples of the sea turtles, you measure them, and you release them back into the environment and recapture them again to see that. You can see the different years here. So why is FP a problem? Well, as you saw, there's giant tumors that develop from this disease. So this can affect their buoyancy and hydrodynamics, also with their swimming and feeding. So if there's a giant tumor on their mouth, they can't really eat properly. Also, if it's on their flippers, they can't swim, they can't get up to the surface correctly to breathe. It can also affect their sight. You can see there's an actual tumor growing on the cornea of the sea turtle's eye. And if they can't see, they can't feed, they can't swim properly, also affecting their predator avoidance. And also death in extreme cases when they're internal growth. The etiology of this disease is most likely a herpesvirus, and we say most likely because it can't currently be cultured in the lab. As Liz was saying earlier, a lot of viruses, we have that problem where we can't culture it. There's also evidence here, so we have transmission electron microscope showing in the top right-hand corner of the tumor development on healthy tissue of the sea turtle. Now we can't really do this anymore because sea turtles are endangered and threatened, so we can't look at those different things and collect samples from the sea turtles very much anymore. There's also ballooning degeneration of the cells growing at the bottom right, and you can see the viral particles growing inside those tumor cells. We can also test the DNA by using PCR and see that viral DNA is present. So why herpes virus? What is that? What are we talking about? Well, herpes viruses are usually very large in genomes, anywhere from 130 kilobase pairs to 165, which is very large, and it's a double-stranded DNA virus. There's also 3 different types of this virus, and there are alpha, beta, and gamma, and they change differently between hosts, how they're cultured, and also their DNA structure. They also evolve with their host, which is usually very slowly, especially in sea turtles. Sea turtles don't evolve, they don't grow like extra limbs and things like that. They're very old animals, so they stay pretty much the same. And there's lytic and latent phases. So in the lytic phase, virions are produced over and over again in the cells, exploding out and infecting other cells over and over again. While in the latent stage, it replicates with the cell in episomal form, so you're not actually seeing that virion coming out. So the possible transmission of this is possibly through the seawater. So it's been seen before in other viruses similar to this, that the virus can survive in the seawater. Also, possible transmission could be leeches. As you can see in this picture here, yes, leeches are found in the ocean. They're not only found in freshwater environments. They do look a little bit different, more worm-like, kind of disturbing. They love to go on these tumors because they're vascularized, so they get the blood flow, get to bite them, and go to a next prey. Also, possibly turtle-to-turtle interactions. This is the most unlikely just because turtles aren't usually aggregated around each other, only for breeding and things like that. So the objective of this whole presentation is to look at all these different species in Florida. So the green, Kemp's Ridley, and loggerhead. All around Florida, look at them, compare their different CFPHB. And also in Hawaii, compare Florida to Hawaii species, looking at that CFPHB as well. And in Hawaii, old versus new DNA. So the OG of CFPHB, in case you don't know, original gangster of CFPHB, which is the Chelonid Fibropapillomatosis-Associated herpesvirus, which is a really short version. Chelonid Fibropapillomatosis-Associated herpesvirus, yes, CFPHB. So the first actual sequence of that was the Ackerman sequence, and it was over 132,000 base pairs, which is fairly large for a virus. And it was in the glottis tumor of this sea turtle, and the glottis is actually in the throat. Now, it's going to show you all a nice picture, but it's really disturbing because sea turtle throats are very clawy looking. If you want to look, go ahead and Google it. Don't want to show you. And that's different because in Hawaii, we see these glottis tumors, but in Florida, it's not actually prevalent at all. We never see the glottis tumors, only in Hawaii. And it's also an alpha herpes virus, and down here below you can see the structure of that genome. So first we have a repeat section, a unique short sequence, another repeat section, and then a unique long. So how do we get this DNA? What do we do with it? First off, we get the tumors from veterinarians. They actually laser remove the tumors off sea turtles. I actually only saw this one time. And then we extract that DNA using a tissue sample extractor. And then we design primers for 5 kb amplicons, and if you do the math, it's quite a bit of primers, especially with all the different sea turtles that we have to sample. And then after that, we do long-range PCR to figure out those amplicons, and we verify with gel electrophoresis. And if that doesn't work, we don't get any bands. As you can see here, there's no bands at all. We have to change the amount of cycles that the thermocycler goes through, or also the different annealing temperatures based on the primers. And then if that doesn't work and we get too many bands, we can also do gel extraction, which is a really cool, fun outfit, where you have glasses and UV protection, and you actually cut out the biggest bands that we're looking for, around 5 kb. After that, we use Illumina sequencing after we clean the samples, and we scaffold those sequences back onto the reference genome that I talked about before, the Ackerman sequence. And then if that doesn't work, we keep going, make more primers, and keep going through all those processes. It takes a very long time. So right now the current coverage is everything except for the repeat sections. So you may think, oh, that's not that bad, whatever, but it's very important. They're 9 kb in length, each of them, and they're for every single one we're missing. So why can't we get them? Well, first off, the primers aren't working. So the GC content is very, very high. If you've done PCR before, if you've designed primers before, you know it has to be around 50%. Well, these are over 60%, so it's not working, no amplification is happening. So instead, we're possibly thinking about using SureSelect, which uses hybridization probes called baits to actually get that DNA out of the virus, and then also target it with magnets. And the importance of these repeat sections is that they hold the latency genes. So these latency genes are very important in that we can see when the virus is replicating, when it's not, and usually we're seeing that the latency is happening when the tumors are present. So it's kind of odd you'd think that they'd be replicating while they're in the tumor form. And also those sections evolve very quickly compared to the rest of the genome. So we want to see those differences between locations and between the different sea turtles. So what's the status of this whole project? Well, we're seeing differences between locations, like I was saying before, with the glottis tumor. We don't see the glottis tumor in Florida, we only see it in Hawaii. So we're seeing those differences there, and then also slightly in the genomes. So you can see in these trees here, they compare the capsid gene and the polymerase gene. You'd actually think that the capsid gene would be more divergent because it's on the outside of the virus, while actually it's showing that the polymerase gene is more divergent, and you can see that by the length of the tree. And you can see that Florida is more closely to itself, and then of course Hawaii. And there is a divergent one right there, loggerhead, and that's because it's a different variant of the virus. Also, we really want to see those repeats because it would show us so much more, so many genes, that we can actually look out and make these trees if we have those repeat sections to see those divergence. So what's the purpose? Why do we need this? Why do we care? Well, first off, there's no standard of treatment for sea turtles right now. Every veterinarian is just doing what they think is best to help these sea turtles. So obviously they give them antivirals, trying to get rid of that virus, but it's not usually working. They can give it to them monthly, they can give it to them weekly, but it's just a very costly thing and it's not working. So we want to help with that. We want to be able to come up with some kind of treatment for them. And you can look at the latency genes to find that out. So if it's in latent form, the antivirals aren't working, and we can compare that to humans and herpesviruses. Usually if it's in latent form, you can't treat a herpesvirus, just like if you have a cold sore or something like that. You can treat it then, but you can't treat it when it's just in your body circulating. And then there's also development of downstream assays. So if we have these whole genomes, we can just look at particular genes instead of the whole entire thing being sequenced every single time, which takes a lot of time. I want to give big acknowledgments to my Breitbart for letting me into her lab, Elizabeth Fassbender for teaching me everything she knows, and of course all the funding and all the other people in the Breitbart lab. Any questions?
So you said you're having problems with getting high DC areas to extend. Have you tried using, well, let me ask, what thermalase do you use? What polymerase?
We use TAC polymerase, and then we've also used the Herc.
Okay, so I recommend using fusion polymerase, which is used for Halophilic archaea. They have GC contents of about 65%. So go for fusion and really high annealing temps, like 72 degrees. And we have really high GC contents on our primers, and that's the only way we get extension. So,
Awesome, thank you.
Any other questions?
Nice job, by the way.
Thank you.
Any correlation with the red tide in southwest Florida or western Florida where your school is located in this disease?
I'm not really sure. I haven't done research on that, but I'll definitely look into it. I want to know more about it just like everyone else does, so I'll look into it.
Do you have any idea how prevalent the virus is in non-clinical turtles? Like, how prevalent is it at a subclinical level? Or do you have any idea because it's difficult to study turtles?
Well, like I said, they are endangered and threatened, so it's harder to hold them in a clinical setting. But yes, they do see it very often. People go out in boats and look at the sea turtles, they see tumors on them. So yeah, it's very prevalent out in the environment, and then also they like to keep them in those closed cages and see how it regresses and things. But like I said, if it's past the 3, it doesn't really get better. It's just after that 3, it just keeps going inside their body, more tumors.
Any other questions? Well, thanks, Makenzie. That was really fun.